All right, welcome to our first lecture video. Just as a reminder, I highly recommend using the study guide that is posted on Canvas. Either print that out or have that up as a Word document. Follow along with the lecture and fill in the study guide as you're doing that. That will help you out immensely in this course. We're going to start out with some basics here, just kind of outlining biology in general, and talk about some key themes and concepts in biology. And then we'll start to get into more of the details of the course in the next few chapters. We're going to talk a lot about the scientific method here. So we are going to talk first about the science of biology in a broad kind of spectrum and talk about it and the rest of what we call the natural sciences. Yes, the natural sciences implies that there might be unnatural sciences. We won't get into any of those in this course, but we will talk a lot about biology, some chemistry, and chemistry is kind of physics. So we'll have a little bit of that in there, but we'll keep the chemistry pretty simple. I know many of you are going to be taking that at some point, so this will be a good introduction to that. Unfortunately, a lot of biology is dictated by chemistry, so we do have to know something about that. We'll talk a lot about the scientific method. How do we actually know stuff and how do we learn things? We'll talk about inductive and deductive reasoning. These are two different methods of kind of trying to explain things and figure things out. And then we'll talk about the difference between what we call basic science and applied science. So we're going to talk about biology, and we might talk about some organisms like this totally not photoshopped picture here. But we're actually in this course going to be diving a lot deeper into the goings on inside of the organisms and some very basic processes. If you go on to take Biology 202, we'll talk much more about the diversity of life that is out there. So we're going to start with this big question here. What is biology? So I'm going to always have some questions in these videos. Why don't you take a second, pause the video, think about what you think biology is. So go ahead and pause the video, think about that. Okay, so what is biology? Well, you probably had a number of very specific examples, but hopefully some of you are already on board with something we're going to be doing a lot of is breaking words down. So we are going to be learning a lot of vocabulary. That means learning new words that often come from Greek and Latin. And when we break up words, they often have roots and bases that have common meanings. So if we break up biology, the first part, bio, is the Greek base for life. Okay, and then tacked on to the end of that is ology, which is Greek for the study of. So it's the study of life. Unfortunately, that leads to a question of what is life. We'll talk a little bit about that philosophically, but we are studying living things in biology. And this brings up a key point. Scientific words seem complicated, many times seem very difficult and foreign to you, but oftentimes if you break them up into pieces, they will make a lot of sense. So that bit of bio, right, we could tack that onto biophysics. So that's the physics of life. That often involves like movement and things like this. So it's really good to start learning these bases. And then when you encounter new words in science and in everyday life, you can often figure out what they mean from that. So when I was in college, I took a course on bioscientific vocabulary. We also have a course here of healthcare related vocabulary type stuff. And that can be really, really useful to take so that you know what new words mean when you encounter them. So here's an example of that. Let's take this seemingly unwieldy term here. So we have here heteromorphic self-incompatibility. Okay, this is something that I studied during my PhD work. Here's some pictures of the flowers that I worked on. We have one mating type called pin, where it's got this long female organ with the anthers, the male bits down here lower in the floral tube, and the reciprocal, the thrum type, has the short female and the male anthers are up higher over here. So you got to cross these two things to get compatible offspring. You can't self-fertilize either. So that is a really complicated system. Now, here's why science has to have really long words. Because it just took me probably a minute to a minute and a half to explain what's going on in this system. If every time that I wanted to talk about this, I had to explain what is going on, that would make for very, very long papers and very, very long conversations about science. So scientific words often have very short, compact kind of terminology that describes something really, really big. Otherwise, we'd be constantly going on, the thing where we do the thing and that thing happens and then this thing happens and that, it would get unwieldy. So heteromorphic self-incompatibility. That describes a whole phenomenon in a short little term. What is that? Well, let's break it down. So let's start with the first part. Hetero. Hetero means different, okay? Different. And then the second part, morph. Morph means form. So heteromorphic means different forms. We have two different forms here. And then we have self-incompatibility. They are incompatible with themselves. They have to have two different forms here. So that word, when we break it down, hopefully makes a little bit more sense there. Okay. So I'm going to try and commonly do this throughout the course. If you're ever unsure what the different parts of a word mean, look them up. It'll help you in the long run. Ask a question, and we will make sure that we're learning this new vocabulary in a way that will help build our repertoire of words here. All right, so back to what is biology. Biology is a big encompassing science, and we're not going to talk about the whole spectrum of biology. We're going to focus in on a few key pieces. So I'm going to show you an image here, and this image kind of puts nature into different scales here. And the first week here in lab, we will work on learning the units of this scale, the metric system, right? But we have a scale of things that we find in nature. And that scale goes from very, very small to very, very large. The observable universe at one end down to subatomic particles at the other end. Biology tends to focus kind of in this spectrum here, although we're influenced by everything in here. We tend to focus in this spectrum here. So that ranges from what you're probably thinking of, right? Organisms like a whale, which is one of our largest organisms out there. Although some people would argue that there are some fungi that like extend for hundreds of kilometers. Those might be the largest living organisms. It's really hard to tell. But when we think of an animal, the whales are the largest animals living today, of course. Dinosaurs were a little bit bigger. Something you might care about, because hopefully you are one, a human. And then inside of a human, you might have an organ system like the heart. You might be interested in smaller things. Fruit flies are on here. We'll talk about why fruit flies are on here. They might not seem very interesting to you, but they are very important in this course. Frog eggs. Then we get into our microbes. So if you take something called microbiology, let's break that one down. What does micro mean? Micro means small, and here's the Greek letter mu, which stands for micro, so these are micrometers here. Anything that you can't see with the naked eye without magnification, that falls into microbiology usually. So something like a paramecium, which is a eukaryotic organism, down to something like a bacteria, which is a prokaryotic organism. We'll talk about what a prokaryote and a eukaryote are. Individual cells. We will learn a lot about cells in this course, and something called organelles that eukaryotes have, down to something that arguably is not living. We'll talk a little bit about this, but viruses are not classified living for various reasons, although they do involve biology, which makes them a little bit odd. They are some of our smallest particles here that involve multiple what we call macromolecules. Then we get down into our molecules here. Proteins, DNA, these are on the small scale. This is kind of the range we're going to talk about here. We will talk about the organismal level a little bit, but in this first part of the biology series, we're actually going to focus a lot on cells and what we call our biological macromolecules here. We will need to talk slightly about atoms, but not too much. Atoms obviously make up molecules, but we will talk about them here. So that is our kind of scale that we're working on here. Now, let's think about various different aspects of this, okay? So when we talk about the study of life, what do you think about in terms of that? What interests you there? That's something that I want to know because I like to tailor this course to what we like. But when I think about it, there's a lot of different levels that are in here. Obviously, being human beings, we often think about medicine, which involves the study of disease and disease-causing organisms. So that might involve microbiology. Something like ecology, you might be interested in that. How do all the different organisms in an ecosystem interact with one another? Something like biotechnology. How do we utilize biology to do things? You probably think of biotechnology as super high-tech, modern, cutting-edge stuff, but I'm going to tell you throughout this course, we've been doing biotechnology for a long time. If any of you have ever baked or brewed or fermented beverages, those are all forms of biotechnology. Okay, food, growing crops, farming, that is biotechnology, right? We've taken crops and we've bred them to be more nutritious and disease resistant and drought tolerant and things like that. There is, of course, the biotechnology aspect of using specific things like DNA and proteins to do stuff. We will learn more about that later in the course once we have our background down. We can have forensics. You're probably interested in that. And things like landscaping, gardening, things like that. All of these are technologies, but biotechnologies and involved in biology here. So we need to know about life for those. But I'm going to take a moment and I'm going to back out a little bit more to give us a bit more of a sense of probably something that you as a potential STEM major are going to need to know something about each of these little things here. So we've got this chart here, and it's kind of got all kinds of different what we would call natural sciences. Natural sciences relate to our physical world and the processes that are going on in there. Now, you're going to want to know, is this chemistry, or is this biology, or is this physics, or sometimes those lines blur. Common problem in science is we like to group things into nice, neat little boxes. And the reality of the world is it doesn't always fit in nice, neat little boxes. So sometimes it's really hard to define the different scientific fields. We tend to break it up into like the life sciences. That is stuff that involves biology. That can get nittier and grittier to things like biochemistry, where often you're not working with too many living things, but you're working with the proteins and the DNA and enzymes and stuff like that kind of are made by living things. The physical sciences, this tends to involve inorganic things like rocks, geology, chemistry, physics, astronomy, things like this. Obviously, though, if you want to study the Earth has been shaped drastically by the things that live on it. We'll talk about photosynthesis. The oxygen in our atmosphere wasn't there when Earth was first formed. Photosynthetic organisms have produced that and enriched our atmosphere in oxygen. So oftentimes the physical sciences are involved with the life sciences as well. Oftentimes, natural sciences are highly quantitative. We like to count and measure things and look at that. So basically, what are the unnatural sciences? I'm not going to call them unnatural sciences, but what are things that aren't natural sciences? Oftentimes, fields that have anything with the word science after it. So this is my biologist dig at the social sciences. We have things like psychology. We might not call a natural science sometimes. Things like political science, right? Those kinds of things don't fall into our natural sciences. Although if you're studying people, they're part of biology. So again, the lines blur there. So we're going to learn a little bit about some of each of these things, mainly some chemistry that goes along with our biology in here. Okay, so now that we've kind of defined what our scope is here, we have a bigger question here of how do we actually know stuff? And you might think that, we know things by reading and doing stuff, but that's not really how we know things, right? We have to have a deeper process to really understand our world. And that is the goal of science. And there is a process to doing science, and there are correct ways to do it, and there are incorrect ways to do it. So science is basically knowledge that covers general truths or operation of what we call general laws, especially when acquired and tested by the scientific method. So the scientific method is key to understanding our world, and that's how we know things, is through the scientific method. We'll talk about the details of that in just a second. So science, the word, comes from the Latin scientia. I apologize, I never took Latin, so my pronunciation might be poor, but that means knowledge, right? So science is knowledge there. And to gather that knowledge, we need a specific process here. We call that the scientific method. Scientific method is a method of research with defined steps that include experiments and careful observation. So we're going to talk about a few key terms here, and we will learn how they are involved in the scientific method here. So we're going to talk about a hypothesis. What is a hypothesis? A hypothesis is a statement that is proposed to explain how a phenomenon works. Okay, let's put a bit more of a real-world example on this. Okay, a hypothesis would be plants need water to grow. That's a hypothesis. That is a statement, right? It's not a question. It's not, what do plants need to grow? A hypothesis is a statement. It is not necessarily correct or incorrect. We have to test the hypothesis. So we set up this hypothesis, and then we have to go and test that. So we'll come back to this experiment, which is a process for testing hypotheses. We'll come back to that in a moment. Let's think of this, do a little practice here. All right, which of these two statements is a hypothesis? On the left, I have, what will happen if I hit a banana frozen in liquid nitrogen? And on the right, I have, if I hit a banana frozen in liquid nitrogen, it will shatter. Okay, so which of those is a hypothesis? Well, the dead giveaway is that there's no question mark after here, right? This is a question. This is a statement, and hypotheses are always statements. So now that we've set up that hypothesis, we have to test that. Unfortunately, I don't have any liquid nitrogen here, so I'm going to pull up a YouTube video of somebody testing this hypothesis for us. All right, so obviously this is someone that has liquid nitrogen on hand. Difficult to store, dangerous to work with, although interesting. So we have our liquid nitrogen here. We have a banana that we're going to stick in the liquid nitrogen. And as you can suspect, liquid nitrogen is very, very cold. And we're going to take that banana, and you can already see something is happening to it, but we have to test our hypothesis here. So going to take a hammer and it shatters after we put it in the liquid nitrogen there. All right, so not probably a revolutionary experiment in your mind because you know some knowledge of the world that has been gathered through other experiments, and the results of those experiments have been communicated to you, right? You know liquid nitrogen is really, really cold. You know that things, when they're put in really, really cold, get brittle. You've probably experienced that in your own life, right? And you know when you hit brittle things that they shatter. Okay, but still, we have to test these things, right? If we want to put together a hypothesis, we have to test it. And the hypothesis there is tested, and we gather data. That data is evidence, right? We often call that evidence. That evidence is going to either support our hypothesis, or we could gather data that rejects our hypothesis. Say that it didn't shatter, say that it got all mushy and squishy. That would reject our hypothesis. In this case, though, our data supported our hypothesis. For complicated philosophical reasons, we never say that we prove hypotheses. Okay. Well, why is that? Well, technically, we cannot do proving because proving means that it will always happen that way. And the thing about science is that we can't test something an infinite number of times, right? We can repeat experiments, but we can't test it infinitely. And we'll come back to this, but there is the possibility that we could get new data that would reject a hypothesis at some point. We'll come back to that in a moment. Okay, so let's kind of test ourselves here, right? What are some hypotheses that this experiment here could be testing? Go ahead and pause the video and think up some hypotheses here. Okay, so what do we have going on here? Well, we have an experiment. We're kind of working backwards here. We're setting up a hypothesis that could be tested by this experiment. Some of you probably went this route. Water is necessary for plant growth. Okay. Others of you might have thought light is necessary for plant growth. If we wanted to get really complicated, we could also go water and light are needed for plant growth. Generally, we try to keep our hypotheses like as reduced as possible, but sometimes we want to test big things, big questions there, and we set up more complicated hypotheses. So we could say both water and sunlight are needed for plant growth. Okay, now we got to set up the experiment. What are we doing here? Well, we have a couple of aspects here. We have our plant, okay, and we have it in soil. You could address the soil aspect too, but this all has the same thing here. And we want to see our light and water needed for growth. So what do we do to test that? Well, the first thing we really need to start with is we need to make sure that our plant will indeed actually grow if there are both light and water there, right? It would be really silly to be testing this if our plant was already dead, right? So we set up what's called a control. A control should show a positive or negative effect. We'll talk a little bit more about this in lab when we set up some experiments. But here, our control is our plant, and it is given both light and water. And we should get a positive result out of this, right? It should grow. If it does not grow, there is something wrong with our plant, potentially. Or that's not what plants need. We're then going to test whether light by itself is enough or whether water by itself is enough. So we leave one plant out in the sun, no water, and we put one plant in the dark, but we give it water, right? So we'll gather data from this experiment, and we could kind of say from this that neither water nor light are sufficient by themselves for plant growth. We need both of these things to occur. Okay, so that's testing a hypothesis. As you can probably imagine, some hypotheses are easier to test than others. Some scientific fields, it's much easier to test a hypothesis than others. So we'll talk a little bit more about that when we talk about why don't we do a lot of research on humans directly. There's a lot of issues with humans, and we can't do experiments easily with them. But let's continue on with the process of science here for now. So after gathering data from lots of experiments, we've got to do lots of experiments, we compile that data together. And if we get enough of a picture from that data, we can form a bunch of different experiments and hypotheses into something that we call a scientific theory. Okay, some people have problems with the terminology theory. You've probably heard it's just a theory. Scientific theories are a little bit different. We have a large, well-substantiated explanation for some aspect of the natural world and the universe around us. So what are some scientific theories? Well, we have the theory of gravity. That is that mass basically attracts things to it. The more mass you have, the more it is attracted to it. We don't have the full answer of why that happens, but we have a theory of gravity that is proposed that objects are attracted to objects of larger mass, right? That is why the apple falls down towards the earth, because earth is big, the apple is small, but earth pulls that mass towards it. Okay, I've never heard anybody say, well, I don't think gravity exists, because gravity is just a theory, right? We've done a lot of experiments, and we have a lot of good data that shows that gravity is there. That being said, we have some possibilities here. I'll get to in a sec. Okay, one that is more contentious with people, and where I have heard evolution's just a theory, is the theory of evolution. Somewhat proposed by Charles Darwin, but he actually probably didn't have a ton of experiments. He was kind of hypothesizing, and after that, people have done a lot of experiments. A great experiment here that can be done in a rapid time scale, right, is looking at microbes. Okay, you've probably heard of antibiotic-resistant bacteria, or superbugs, sometimes they're called. There, right, we have a population of bacteria. There's millions or billions of bacteria in the population. Through genetics, some of them are going to have mutations, and if we apply antibiotics to them, some of them might be resistant to antibiotics. Okay, and those ones that are resistant will survive and reproduce, and the population will change to mainly antibiotic resistant at that point. That's evolution in action. That's evolution in action that affects a lot of us, right? You've probably heard of MRSA, methicillin-resistant Staphylococcus aureus. That is a bacteria that is resistant to a common antibiotic, and it causes lots of problems in hospitals. To test evolution on a larger kind of time scale, we take evidence from fossils and other experiments that are looking at more broad things, how different populations have changed in different locations, things like that. So Darwin probably didn't do a lot of these experiments, but a lot of people have done a lot of experiments afterwards, and we've established the theory of evolution, and we have a great mechanism for it. We're going to talk all about it in this course. It's called genetics. Okay, so whereas gravity, we can't fully explain why gravity happens. There's still a lot of parts that are kind of mysterious to us. Evolution, we have a great mechanism for it. That being said, we use the term scientific theory because science should always be open to new data. That's why we say we don't prove things. Because if new evidence came to light that suddenly gravity doesn't work anymore, we'd have to throw that theory out, right? It's very unlikely that's going to happen, but we have to be open to that as scientists. So that is the philosophical reason we use the term theory, and we don't say the word prove here. Mathematicians can prove things when they write out mathematical proofs. We support hypotheses in science. Okay, so how do you actually go about doing this? Well, we have this process called research. Unfortunately, everyone learns this when they go out and do some science, right? So I learned this the hard way as an undergraduate in grad school. And my boss said it's called research, not search, because you got to do things over and over again. You got to keep doing it. So we're going to go out and observe the world, and we're going to why does this happen, right? Why is the sky blue, or why is the grass green? Or you start with a question. Okay, from that question, usually what you do as a scientist is you go and read, because somebody else has probably had that question before. The really interesting ones are when you read a lot and you realize no one has asked that question or no one has answered that question satisfactorily for me. So we then form a hypothesis that will hopefully address that question. We make some predictions based on that hypothesis, and we do an experiment to test that, and then we analyze the results. If the results support our hypothesis, we usually repeat that to make sure that it keeps happening over and over again. If our results did not support our hypothesis, we try again and we modify our hypothesis. Either way, you're doing this thing over and over again, and more often than not, you go this route. There's a lot of failure involved in science. A hypothesis, therefore, has to be falsifiable. There should be a way to show that it is incorrect. This is where science and the supernatural don't quite gel. If your explanation is that aliens came down and changed it, you got to be able to prove that, right? And I just said prove. You got to be able to show evidence that supports that. If you can't, we have to throw that one out, right? So we need our hypotheses to be falsifiable. So we can't just say, well, it's magic. Sometimes it works and sometimes it doesn't. We really want to drill down to something that we can address and support well. So we're going to talk a little bit more, especially in lab, about our kind of experiment part of thing. And experiments have multiple aspects to them. We have variables. These are things that change. There's actually multiple types of variables. And we have controls. Controls are important to show that everything's working properly and that the effects that we observe are due to changing variables, not just the process of doing the experiment itself. Right, so we saw that in our experiment here. Water and light together were our control. In more detailed terms, this would be a positive control. So if that's a positive control, let's think about what a negative control would be. A negative control would be key here too. Negative control would be neither of these things, right? No water, no light. We're pretty sure that plants will not grow under those conditions, right? So negative is no effect. Positive is showing some effect, right? And then we're testing the different variables, light and water, in these two examples here. Okay. Scientific reasoning can take two different tracks. We're going to briefly talk about this. I don't want you to get too hung up on this, but I want you to think about this more broadly, because going forward in your professional careers, you might be involved in either aspect of this or both aspects of this. So scientific reasoning involves logical thinking. You can take whole courses on logic if you go into computer science or something like that. You definitely will take formal logic classes. But we have two different types of reasoning here. We have what's called inductive reasoning and deductive reasoning. And these differ slightly. So let's talk about inductive reasoning. This uses related observations to arrive at a general conclusion. Okay, so you're observing something and then drawing a conclusion from what you observed. We'll come back to that. Let's talk deductive reasoning. Using general principle or law to predict specific results. So this is a different direction, right? So in inductive reasoning, I'm observing a whole bunch of things. Like over and over again, I observe 20,000 birds, and then I try to draw a conclusion from that. In deductive reasoning, I'm saying, I know gravity exists, I know how thermodynamics work, which is study of temperature and stuff like that. I know how chemistry works. I can use those laws of gravity and how chemistry works and things like that to predict what the result might be. I'm going to build a hypothesis on that. So these kind of approach it from different perspectives. So descriptive, we're trying to infer a conclusion from many observations. We're doing correlation. To do that, we're going to need to use statistics. So if you're going to continue on in science, at some point you're probably going to need to take a statistics course. It'll be very useful for you, particularly if you're going from the inductive side. Deductive reason, well, you probably should know some statistics as well, but we're starting with a specific question and devising hypotheses, and then using our natural laws to kind of make predictions to inform our hypotheses. Still got to go and test those, though, right? Rarely do scientists use such formal language, and rarely do they do one versus the other. It's generally a mishmash of the two things. I have never sat down with anyone and been like, let's use deductive reasoning today. So some of you might be old enough to remember this show, which does not represent how normal scientists talk. So I'm going to show you a little clip here. All right, this clip is from the show Bones. Some of you may have watched this. My wife really likes this show, so I've watched every episode of it. I enjoy it as well. But here we're going to see some scientists using some language that probably doesn't get used in real life here.
You won't have to.
I entered all the modern and historical city plans, including ventilation shafts and tunnels, plus the newer schematics.
There's also oral accounts of tunnel construction and underground passages.
Oh, wow. All that exists under the city.
Yes, but we can corroborate.
Good point, Bones. Not rushing off to find those guys.
How accurate is this?
Blue is modern, near 100% accurate. Yellow is historical.
Estimate 80%.
Red represents less exact renderings from stories, memoirs, accounts from city workers.
Fortunately, if this treasure exists, it probably exists in one of the red tunnels.
Oh, we found that Civil War victim near Cave-In. Maybe the treasure's on the other side.
Inductive, reductive, or deductive?
Deductive.
As you wish. Ms. Montenegro, please remove all tunnels containing power, cable, or utility lines.
And fiber optics.
Yes, also steam tunnels and transit access.
Oh, what about diamond dust? Well, you said that there was diamond dust in the old tunnels. There was also diamond dust on the Civil War guy. So, what? I'm not allowed to help now?
That's inductive logic.
We agreed on deductive.
I'm sorry, I'm just, trying to think outside your box.
All right, so in this clip, right, they agree on a formal version of logic that they're going to use, and Booth, being the lowly FBI agent, doesn't understand that, and they're trying to deduce by removing certain things that are charted there where this treasure might be. He's trying to use inductive reasoning. He knew that diamond dust was on there. What about that? Why might that be coming from? He's using an observation that they have there. I've never had a conversation like that. So this distinction between the two types of logic is not super critical, but sometimes it can be useful to use. If you're doing astronomy, it's very hard to do a large number of observations. Sometimes you have to use deductive reasoning to simulate things or something like that. Correlations, right? Inductive reasoning uses correlations. Maybe I show you this delicious extreme grilled cheese here, and we map your brain, and we do that for hundreds or thousands of people, and whenever they see the delicious grilled cheese, this part of their brain lights up, and we might induce, using inductive or correlate, that this part of the brain is the grilled cheese center or whatever, right? It responds to grilled cheese like this. Whereas deductive reasoning, we might be using something like simulations to predict what's going to happen later using a computer model. So these are the historical data, and we're trying to predict if climate change occurs, temperatures change, things like that. How might our plant hardiness zones change as well? These are based on what plants can grow in what regions. If you garden at home, you might have consulted this, because in East Idaho, we're kind of like in this weird area where it gets really cold and it gets really hot. So our hardiness zone can be weird. I worry about this with my lawn, right? What type of grass is in my lawn? Can it withstand the high temperature in the summer and the cold temperature in the winter? If the climate shifts, are these hardiness zones going to shift? Maybe they'll move further north, right? So we might want to know that. You might want to know that if you're a farmer, based on what crops you can grow and what money you can make. So how do these two things work together, and how can we do things with them? Well, let's take the example of seed burrs. So I'm sure if you've ever gone hiking, you've gotten these on you. They get on your socks, they get on your pants, right? They're little seeds that stick to you. And I'm not going to get into the history of this too much, but the book does talk a lot about this example and the scientists involved in it. But we have this observation, right, that burrs stick to a dog's fur. And we might want to know, why do they do that? So the hypothesis is that they have microscopic hook structures which stick to the hair, right? That's kind of based on some observation, but really to test this hypothesis, we need a microscope. So here is an example of that under a microscope. Except, I kind of lied to you, this isn't actually the burr sticking, the fur and the burr sticking to it, although it looks like it. This is an example of where biology has led to something that we called applied science. So we did basic research. We're like, hey, why do the burrs stick to the fur? That's our basic research there. But then we might want to apply that to something. So those of you that are maybe less fashion conscious and more into utility, you might wear Velcro shoes, or maybe you've used Velcro in other capacities. Velcro is directly inspired by that natural observation. Velcro has a bunch of tiny little hairs, essentially, or furs, and it's got a side with a bunch of little loops on it. So it's often called hook and loop, and just like the burr, the little hooks stick to the little fur. So here we've observed the natural world, and we have developed something that's very, very useful, although maybe not the most fashionable thing. Very, very convenient, right? So this is part of the scientific method, using our findings to make technologies for us. And this scientific method, there's a lot of history behind it. It was first kind of formally stated by Sir Francis Bacon, famous scientist. You can learn all about him in the scientific method. There's lots of books on this subject. But I just want you to be thinking about this, right? Knowledge has to be based on hypotheses that are tested and then supported through data. And those results have to be reported. If you take upper level science courses, you will read primary literature where this process is described. If you take my Microbiology 250 class, we read some primary papers where we look at this whole process in detail. All right, so I'm going to give you a practice problem here. This practice problem involves my toaster not working. Okay, so we have a scientific method here. We have some things that I want you to match to the different parts of the scientific method. So go ahead and pause the video and think this through. We start here with our observation that my toaster doesn't toast bread. Okay. So we have an observation. That's the start here. We might then develop a question, right? Why doesn't my toaster work? That's probably the question you want to know the answer to. Okay, to test this, to examine this question, we need to form some hypotheses and then test them. So, one hypothesis is there is something wrong with the electrical outlet. Okay, that's one hypothesis. You could have other hypotheses. My toaster is burned out, or the electricity is shut off in my house, right? There's a lot of different hypotheses that could be made. This is the one we're going to test first, okay? So there's something wrong with my electrical outlet. This is kind of the is it plugged in kind of one, right? Like, okay, so that's our hypothesis. We need to make an experiment to examine that. So we are going to make a prediction, okay, of what should happen. If something's wrong with the outlet, my coffee maker also won't work when plugged in. Okay. Of course, we're not sure that the coffee maker works either, but we believe it does. It would be unlikely that two of our appliances would fail on the same day. Totally possible, right? But we're going to plug the coffee maker in. So that's our prediction. We're going to do the experiment. I plug my coffee maker into the outlet. What's our result from that? What's our data we gather? My coffee maker works. So what does that do to our hypothesis? Remember, our hypothesis up here was there is something wrong with the electrical outlet. What's our data suggest there? Our hypothesis is not supported here because my coffee maker works. So what do we got to do? We got to go back and revise our hypothesis. Probably to my toaster is not working because it's broken, right? Like, what is broken? Well, you'd have to form another hypothesis if you wanted, or you just say, I don't care why it's not working right. I'm going to go buy a new one. Okay. But you could test the fuse. You could test, all different aspects. If you really got in there with a voltmeter and stuff like that, you could get into which part is broken in my toaster. So that's the scientific method in action right there, right? You probably use this when you go through logical problem solving. In science, though, you would write all that down and test it multiple times, different outlets, things like this, just to make sure. So we talked about the difference between what we call basic science and applied science, and I showed you the burrs and the dog's fur as an example of that. I want to give you another example that's actually more applicable to things that we're going to study in this course and, for reasons, local kind of story to this. So our basic science is just to do science so that we learn more, right? It's to expand our knowledge, regardless if there's a short-term real-world application. This is a big problem in science because you have to tell people, hey, I need money to just learn more things. Okay, that might sound perfectly normal to you, someone who is trying to learn things, right? You're like, yeah, I want to learn stuff. And that's cool. But to everybody else that you're asking money from, they're going to be like, why? Why am I going to give you money? How is this going to benefit me, right? And that's tough because basic science doesn't really address that real-world application. That's the part of science we call applied science, technology, using science to solve real-world problems. But I'm going to show you an example of where doing basic science, even though we didn't know how it would be useful, has paid off to both make our world better and make some people a lot of money, which maybe you're interested in. So money, unfortunately, is a big part of science. And we kind of group this into what we call industrial science. Industry tends to do applied science, and academia, the university side and the basic research side, is often more basic science. All right, so we're going to examine this example here on basic science and applied science. And we're going to go to some place that I really think is cool, Yellowstone. And hopefully you've been to Yellowstone. If you have, you probably saw some of the pools and the features. Grand Prismatic is the most famous, of course, but we have this variety of colors that we can observe in the Yellowstone hot springs. Big question, what makes those colors in the Yellowstone hot springs? It's like real unique, right? We rarely see this beautiful rainbow color in other areas. Why is it happening in Yellowstone? So our basic science was conducted primarily by this guy named Thomas Brock. This is him much later, but in the, I think it's 50s and 60s, he went to Yellowstone and he collected samples of the water and put it under a microscope. And lo and behold, there are bacteria and these other things called archaea that we'll talk about. They're single-celled organisms, but they're not grouped with bacteria. They're separate lineage. One of those bacteria, though, is called Thermus aquaticus. Okay, sometimes scientific names, the genus and the species, make sense. Thermus means hot, and aquaticus, aquatic, means water. So this is hot water bacteria. Makes sense here. Sometimes we name things smart. So, cool story, bro. Like, you might be interested in this, but the general public's like, why am I paying this guy to go find tiny microscopic organisms in Yellowstone? Well, maybe they'll make a cool PBS nature documentary on it, but like that doesn't make me any money. Why do I care? Well, here's where science and knowing a lot of things about science and knowing a broad thing, like broad width of information, is really critical for science, because the biggest scientific discoveries and inventions come when you know lots of things and you can connect two very different topics together. Okay, when you make connections, you make history, basically. So we're going to go shift over to the applied side. Okay, this might not be clear why it's connected yet, but in the early 80s, we had this big question. We knew DNA was the genetic material, and we wanted to study it. And one thing that we needed to do was we needed to be able to replicate DNA, to make copies of DNA in a test tube. If you want to work with it, you need lots of copies. So this guy, his name's Kerry Mullis, he was working at a company that was interested in figuring out ways to do this. So we knew from chemistry that if you heat DNA up, and we're going to learn all about this process, DNA will unzip. It will open up. And this is what happens in our cells, not by heating it up, but by another process we'll learn about. So if you unzip the DNA, you can then add these short little pieces of DNA called a primer, and you can add this enzyme, which is a protein that is made by living things called DNA polymerase. DNA polymerase is an enzyme that makes polymers of DNA. It copies DNA. So if we start with one DNA double helix, we go through this process that we call polymerase chain reaction, we can end up with 2 copies of that double helix. Okay, and you do that again, you get 4 and 16 and so on and so forth. You can get a lot of copies in a short amount of time. The problem is, to get this unzipping to happen, you need to heat it up to about 95 degrees Celsius. Now, if you're not familiar with the metric system, we'll do a little bit of this in the lab, but water boils at 100 degrees Celsius. So we need to heat the DNA up to almost boiling. Okay. Now, why is that problematic? Well, the problem comes with this enzyme here, DNA polymerase. DNA polymerase is an enzyme. That means it's made out of protein, and most proteins will denature, or basically unfold, when they are exposed to heat. You probably have experienced this if you have ever cooked an egg. The egg white is liquid. It has proteins in it that when you heat them, unfold or denature and become solid. Okay, you probably have never tried to uncook an egg, but let me tell you that this denatured protein will not go back to normal, right? If you let it cool down, it won't fold back up. DNA polymerase is an enzyme. It is a folded protein. It does its job of copying DNA. If we take that DNA polymerase from us, humans, or something like E. coli, bacteria, which is very common, it's easy to get enzymes out of them, if we stuck ourselves at 95 degrees C, what would happen? We would die. Part of that dying is because our proteins denature. Same thing with E. coli. So if we're using E. coli DNA polymerase, it's going to denature when we heat that DNA up. Kerry Mullis made the connection between the Thermus aquaticus, which lives in hot springs that are almost probably 95 degrees C. He hypothesized that they will have evolved to resist that temperature, and their proteins will have evolved to resist that temperature. So he got a sample of this Thermus aquaticus. Thomas Brock had some stored in his freezer. I think he got it from him. He got a sample of it and tested this, and lo and behold, the enzyme DNA polymerase was able to withstand that high temperature. And that enabled him to do all those cycles of PCR without having to add more enzyme, which is very expensive. So making this connection between this hot spring bacteria and its evolution and how that could be applied made Kerry Mullis some money. They paid him about $300,000, his company. That sounds pretty good. I believe they went on to patent that and sell that for $300 million. Kerry Mullis, although he got paid, the company got paid a lot more for that. And that PCR technique you've probably heard of when we talk about COVID testing. PCR is a way to examine whether certain DNA or RNA molecules are present. So we take a swab from a patient and extract the RNA, turn it into DNA for reasons, and then we try to make copies of that. And if you can make copies, then we know that they have the virus. If no copies get made, they're negative for the virus. So we could test all kinds of different microbes and all kinds of things. So PCR, this basic Technique of copying DNA gets used all the time. And we will do PCR in this course, so you will see how it is useful. So this is an example of basic science being applied, even though it was many years later, 20 years later, and people made a lot of money off it. All right, to finish this out, I know this first section is real long, but we're going to talk about how scientific work gets reported and the process of peer review. So we're not going to dive too much into this because this is an intro biology course, but as you go upper level, even if you're in other fields or things like that, you're probably going to read primary literature, which is the first part. Primary papers report the experiments and some interpretation of that data. They're usually very, very narrow in scope, okay? They have some standard parts in abstract, which is like a little summary of it, an introduction, which gives some background knowledge. So if someone is not an expert in that particular part of the field, they can get some sense of what this is talking about and why do this. Critical part, materials and methods. You have to describe exactly what you did so that somebody could replicate your work from just reading this section. That's really critical in the scientific process. The results you observed from your experiment, some discussion of those results, what they might mean, and then you include references, because in the introduction and the materials and methods, you're referring to somebody did this before and I modified it, or things like this, or here's a bunch of background research, and there's generally hundreds of these in there. So that is a primary research article. You don't get to just write this and throw it up on the internet. You really need to have it what we call peer reviewed. And that means sending it to several different people, usually 3, that are unrelated to this work. They did not participate in it. Usually how this breaks down is there are about 3 other people in your field or related field that it gets sent to. One of them you probably know and you probably get on with pretty well. One of them doesn't really care too much about your stuff. And one of them is your bitter rival and hates you and will tear it apart. That's what we want, right? We want to make sure that the science is being done properly. So this goes through a rigorous peer review. And usually you have to go back and make revisions. You have to do more experiments. They say, I don't think this, supports what you're saying. You need to do this, or you forgot about that. And then you have to revise it. And usually like a year later, it ends up getting published in a scientific journal. Okay, on the other side, that's really small in scope. Sometimes we want to make bigger kind of cohesive things. And these are called review papers. You don't actually do any experiments here, but what you do is you take hundreds of these primary papers and you compile them into kind of a cohesive narrative that tells us about the field. We're taking lots of data and kind of more broadly explaining what might be going on based on experiments. So review papers are generally easier to understand and synthesize our knowledge and current understanding of a topic. All right, that's a big chunk. I apologize. Usually the sections are smaller, so they'll be broken up, but we had a lot to go through in this first piece. So the chapter 1.1 summary, I'll always end with a summary here of what the most important pieces are here. And remember, when you're going through these videos, make sure you use your study guide because the study guide is what's going to be on the test, and that will help you. The way to think about the study guide is anything on the study guide will be fair game on the test, but not everything on the study guide makes it to the test. So there are a few things on the study guide that might not show up there, but anything on the test will relate to the study guide. So we've talked a lot about how scientific words can be broken down into parts to understand them. We talked about biology, it's the natural science that studies life. And we talked a lot about the scientific method, setting up testable hypotheses. Well-designed experiments have variables that change and are tested, and then controls that show that any effect we saw was due to our variables, not something else. And they either provide evidence to support or reject our hypotheses. When we do enough experiments and gather enough evidence, we compile that into scientific laws that describe the phenomena we observe in the universe. Scientists use kind of 2 different types of logic: inductive reasoning. You collect observations and make some sort of correlative conclusion. Or deductive, you start with general principles and laws and try to predict a specific result. Talked about basic science, just doing the learning things, doing science to learn new things, and then applied science using that knowledge to solve real world problems. Talked about publishing, primary papers, these report the individual experiments, and review papers kind of take many of those primary papers and collate it all into one cohesive unit. All right, that ends chapter 1.1. Chapter 1.2 will be much shorter, so go ahead and move on to that when you have time.